Yes. Good morning, everyone. We will discuss about Mendel's laws or laws of inheritance. And that's done mostly the knowledge. I hope you have watched all the videos that I sent you yesterday. If you have sent it, it is all redundant. I'm only trying to make up some things here and there so that you will be able to have a better understanding. Those videos, I think you would find them, they are well made. It's called from the channel Don't Memorize. They have many other concepts. Even if you have siblings or somebody elders, you can also try to suggest these videos to them. Yes. So Gregor Mendel was a monk, um, and he was working with church and other stuff, but he's also doing experiments. Okay, well, while we see many of the people from times a little older, I mean, ancient people, many of them had different professions. Somebody was selling buttons and then invented microscopes. Somebody was a lawyer, but does something about botany and so many other things. You have a mixture of people where, unlike in these times where somebody has to pursue one thing and there is not as much flexibility. Well, the point is, there are some things fantastic about Mendel's, because the way he has deciphered. I hope you understand that nobody could see what is a gene. Nobody could actually imagine that. So many of the theoretical foundations, like elucidating the structure of DNA, or thinking about evolution by Darwin or Mendel's laws, these are all some things which requires real mental capacities. In the sense, there are two things that are required. First is intelligence, something I just with spelling, and the other one is technology. Okay. In olden days, people who were constantly thinking about something, that is using their intelligence, would become scientists. Nowadays, if you see in the modern scientists, majority of them are dependent on technology. I think some people usually call them also as technicians and not scientists. Okay, but everything is science, so it's all right, I think. Either what makes you different is here in the case of intelligence, you are the one deciding what will happen. I mean, whether you'll succeed or not. In this case, if the technology is available, you are able to do something. If the technology is not within your reach, then you cannot do something about it. Right? You are dependent on technology here. Whereas in this case, you are dependent on your intelligence, your learning. I think everybody should focus on improving their own skills. First is to get a theoretical background, and then try to improve your own thinking skills. Because technology is there. It may be available to you, it may not be available to you, but this is within your control. So for intelligence, you should start with knowledge, building your knowledge, and knowledge will give you intelligence. Right? And intelligence will get you more knowledge. It's a cycle then onwards. But first, of course, we should know what is already existing, and then based on that, you will be able to use that knowledge, that is intelligence. And because of intelligence, you will learn more things, that is knowledge, and this cycle will keep on going. Okay. I hope you will draw inspiration from many scientists that are there, that have been in our own field in molecular biology, and you have to read a little bit about them to draw inspiration. Okay. So there are some things we will learn about Mendel in a while. But yesterday you have watched some videos. I would want you to tell me some of the terms that you must have heard, and try to keep it in one or two ways. First one I will go is gene, or second I will say an allele. Third is a trait, or fourth, or phenotype. Can you tell me any more? Phenotype. Fourth, you can say as genotype. I'm just trying to build the vocabulary that you have learned in the last from those videos. If you don't understand these terms, then you'll have difficulty in the future. So that's why I'm focusing on that. Anybody else? Homozygous. Then you will have heterozygous. Are there any other terms? Anybody?
So dominant and recessive.
That is right. Yes, dominant. Recessive. Anything else? Anybody? Okay, we will go with these first, and then we'll see. What is a gene? For the simple understanding, we will put it as anything that codes for a protein or RNA. You can just simply say anything that codes for RNA, and we'll go that way. Anything that codes for RNA. You can just say it like that. There are some people have differences in defining what is a gene or not, because some RNAs are sometimes some parts of the genome are transcribed and we don't know if they have a function or not. In that case, should we consider it as gene, or should we just think that by randomly RNA polymerase happened to have expressed it? So we don't know that. But usually we will say that a gene codes for a functional RNA. Functional RNA, when I say, it could be a tRNA, it could be rRNA. What is tRNA and what is rRNA? Or mRNA. Yes, transfer RNA, ribosomal RNA, there are small RNAs, there are microRNAs, other things also. But there are also, these are three important ones. And mRNAs will give rise to proteins. Okay? So a gene is anything that codes for functional RNA. The function being, you can also, I will just add small RNA and microRNAs also, which have regulatory functions. Okay, those are genes. What is an allele? Say, for example, it is an alternative form of a gene. When you say, for example, this is a gene, it has a start codon and a stop codon. Upon transcription, it will give rise to mRNA, and upon translation, it gives rise to a protein. Okay, we will call it a gene A. It is an enzyme that gives some trait. Trait is the character or feature or phenotype. Say, for example, there is another gene which is almost similar to gene A in terms of sequence. I will, for simplicity, I will just write a few sequences for you to understand and to demonstrate what is an allele and what is not. Okay, so I hope you will be able to use your discretion in that. ATG, I should put. Okay, the start codon is ATG usually, so CCGC, and I'll put a GA TGA. That is a stop codon. Okay, so it basically codes for 1, 2, 3 amino acids. That's not a real gene, but for our understanding, I put it like that. Here is another case, ATG, then CCC, and CGA. CGA, TGA. I hope you are able to see that. Okay. If you, I didn't write it clearly, but if you notice properly, you will see that there is a difference. The difference is this particular 1G, and in that location, the other one has C. All the sequence is exactly same, right? Except for the part where this one base is different. So typically this gene is same as the gene A here, but there is a difference, right? Gene A, which is in a different form, and that's A, and I will just erase a little bit. A1 and A2 are alleles, A1 and A2. One can also have different forms. You can have more than one allele also. So I have here A1 and A2 are alleles. So because of the, so basically this will give rise to mRNA upon transcription. It will form a protein. Maybe because of this mutation or change, nucleotide sequence change, it may not be functional properly. Okay, we will learn about these concepts a lot because of, so 1 second. Yeah, the enzyme here gives rise to a phenotype, whereas because the enzyme is non-functional, there is no phenotype. Okay, or there is that character. Say for example, this one gives rise to skin color. Okay, because there is no enzyme, the skin is colorless, right? Something like albinism, people who do not produce melanin and so on. I hope you understood what is an allele. Allele is alternative form of the same gene. Why is it an alternative form? Which means that the nucleotide sequence of this gene and this one, they are absolutely almost same, except for this one base here. So they're not this is A2, okay? And it could be something like this. Because of the nucleotide sequence change, they may have different functions. So that's an allele. What is an allele? Alternative form of a gene. And then we have a trait or phenotype, that is a feature. Here I have used a feature of skin color. You can talk about height. You can talk about everything, anything that we have. Say if you talk about somebody, the person is a happy person, that is a trait also. It might be genetical or not, that's a different. But when you say as a phenotype or trait, you're talking about features and characteristics. Those could be structures. The plant is very tall, or the plant is very short. The plant has many branches. The plant has fewer branches. Something like that. You're talking about structures. You can also talk about behaviors, like the behaviors of bee. How do they find the place where the nectar is present in abundance? How does it communicate it to the rest of its beehive mates, other bees, and so on? It could be anything. You can talk about longevity. How long do they survive? You can talk about any of the features. In some cases, they think intelligence also has some genetic role, and it is thought that it's usually coming from the mother. Okay, but all the traits that we know of, all the phenotypes, or majority of the phenotypes, are based on two things. One is nature. When I say nature here, what it means is the genetic, what is there in the genes, nature of the person, inherent genetic nature is nature of the person, and the other one is nurture. It is to say that how the person has been trained. Say, for example, if somebody, as a newborn child, okay, I'm just making up something so that you'll understand it. As a newborn child, we have genes for formation of brain, right? We all have it. But then we are all different people. We have different tastes, we have different speed of learning, we have diversity in thinking capacity, mathematics or arts, or you can take off anything, everything that we have in our us, what we are unique mostly by our brain. Of course, the body also has limitations and strengths. Somebody can put on muscle very fast, somebody can put on fat very fast. It all depends on metabolism and so on. Here I'm talking about the brain. We all have the genes to make up a brain, and then we have different capacities, different strengths in our different types of intelligence. Somebody is good at sports, somebody is good at mathematics, somebody is good at drawing, painting, singing, dancing. You can take any of them. Why that happens is based on many other things, and that all those factors, the ability to form the brain is nature. What happens and how the brain has modified is determines what happens to our brain, how we change, right? That is all the nurture part, and most of the nurture will be from the home, then from friends, school, teachers, movies, and so on. A majority of the things we develop interest during our school time or early childhood, and then maybe until adulthood we get influenced, and then we keep changing, something like that. I hope you understood what is meant by nature and nurture here, right? We all have brains, but we are all unique in our tastes and capacities and so on. That depends on our focus, what our parents have put us, what kind of friends we have chosen, and everything else like that. Okay, it also depends on what kind of bad influences, like maybe somebody heating us up, or those kinds of things also may affect our brain and our behaviors. Okay, I hope you understood that part. Then we have to discuss about what is genotype and what is phenotype. We all have learned that majority of the organisms are diploid. When you say diploid, that means they have two sets, each set coming from one parent. So I want to use a different slide probably. Just give me a minute. Okay, let us this. Why are we diploid? We have already finished reading about mitosis, meiosis, and so on. If you remember what happened in meiosis, we will notice that we all have, at the end of meiosis, there is a gamete formation. One is a sperm. It has N set, and there is an egg cell, that is oogenesis, and the other one, this would be called as spermatogenesis, and this will be oogenesis, formation of the egg, and it is also haploid. It's not exactly haploid, you will learn the actual details in little more, but for the moment you can consider it as haploid, because when it fuses during fertilization, fusion of these two gametes is called as fertilization. Only haploid set from sperm and one N from egg will make this zygote, and from that zygote we will come up, right? So in all the cells of our body, we have two sets. we are also called as biparental. What does it mean? One set is coming from one gamete, another set is coming from another gamete. So we have two parents, right? Mother and father. So we are diploid, and that means we have two sets of each gene. As I was discussing in the earlier one, A1 and A2 are two alleles. A1 is one allele, A2 is another allele, right, of gene A. What is the difference if we go back? The difference between this one and this one, allele A1 and A2. A1 will produce a functional protein, and that because of that functional protein there is a trait, something like skin color. In case of A2, there is no because of the mutation here, right? There is a mutation because of which it forms a non-functional protein, because of which there is no skin color, or it remains colorless. So remember that. So here we are again discussing the same, that is A1 and A2. Just recollect, A1 will give you skin color, say brown, and A2 no color. Just remember like that. If the parents, if an individual has A1, that means that person, he or she has A1, two copies of A1. We are talking about what kind of genes they have. If we are talking about what kind of skin color, what kind of color is the skin color? If you are asking, that means we are talking at the level of phenotype. If I am asking about what kind of genes, skin color genes are present, then I'm talking about genes of it. Here in the other case, I'm asking how does the person look, brown or colorless? Then I'm talking phenotype. If I say which genes are there, then it is genotype. Okay. Yes, I think I have described majority of them. Okay, homozygous and heterozygous, that is one more thing. If a person has A1, or A2, then they are homozygous. When you have the same alleles, then you call it as homozygous. If a person has A1, A2, then it is heterozygous. Hetero is different, homo is same. Right? So homozygous and heterozygous. Now I would want to also describe what is dominant and recessive. Say a person has A1, okay, or I should actually tell as a person is homozygous for A1. Then what do you get? This allele would have given rise to a functional gene. This allele would have given rise to a functional gene, because of which the person will be brown, right? Instead, if I take a different scenario, A2 and A2, what can I expect then? Then the A2 produces a non-functional protein. So it doesn't produce a functional protein, because of which the person, both the genes are non-functional, so they have colorless skin. And now we have a different scenario where there is a situation like in the case of heterozygosity, A1, A2. Then we will have, this will give rise to a functional protein, whereas A2 allele will give rise to a non-functional protein, because of which this one will give rise to a brown color, and this one does not give to any color. So I will give a numbers to each of them: person 1, person 2, and person 3. Okay, what will be the phenotype of person 1? It will be brown. Why is that? There is a functional protein, functional enzyme, because of which pigment is produced, and so they will be brown. What about 2? The person has genotype A2, they do not, both of them do not produce any functional protein, so they will remain colorless. And if a person is heterozygous, then it's like what will it have? What will be there? It will still be brown, will be the color, because there is at least one copy of a functional enzyme, functional gene, which will produce a functional protein or enzyme that will produce the pigment. So we have brown color here, right? So presence of the enzyme or absence of, so which one is dominant and which one is recessive? That is what I'm trying to get to now. Which is dominant? Obviously, the one that is producing functional protein. It produces the enzyme, right? I mean, the pigment, because of which its trait is visible, even when it is in, even though there is only one copy. Those are referred to as dominant. Whereas A2 is recessive, because if it has to exhibit its phenotype, the phenotype here is being colorless, right? It has to be homozygous. If it is homozygous, then it will exhibit its character, even if they, but not otherwise. Even in heterozygous condition, the dominant one would show its phenotype. Okay. I hope you understood these terms, and we can now move on to some things about Mendel. Mendel has done several experiments using peas, and there are some things mysterious in understanding how Mendel chose the plants, why that plant is chosen, why those traits. He has searched, he has worked on seven traits or so. Is it possible that Mendel has worked on many other traits, but he has come to only these ones? There is some controversy on that, because his readings or findings seem to be too good to be true. That is how some scientists will see. You can go and check on the internet, but that's not, that is still, there is something fantastic about Mendel's experiments because of the experimental approach, because he is dealing with uncertainty. That is, that's very difficult for anybody to do. You don't know what you're going to find. You don't know what to look for. You don't know how to look for. And you are doing some experiments and trying to make keen observations. Dealing with that kind of things will require a lot of intelligence, or actually more of dedicated and focused thinking about the subject. And the other part is he had a quantitative approach that he has taken in trying to determine what is good, what is valid or not. He was able to come up with some things where he could guess the phenotypes based, or genotypes from the based on the phenotypes and so on. So he would usually take plants and the flowers, and he would make several types of artificial pollination. So there are pollen grains, sorry, anthers. In some plants, he would cut them off and take the pollen from other cross pollination. He would do. He is controlling the experiments very well, right? And so those are some of the things that Mendel has done, and that's why it's important. We'll get into the first one is about independent segregation, the law of independent segregation. So he was working on in a trait. This is called as monohybrid cross. You already have done it. In the next class, we will discuss some of the problems. I thought of giving it, but I couldn't make it up. But yes. Monohybrid is, you can think mono is one. You're talking about only one trait. Okay, that's simple to do that. So there is a convention about how you write about what is dominant, what is recessive, and so on. In the earlier case, I have chosen A1, A2, like that. Why is that I chose something like that is one gene may have more than one allele. That means more forms of different forms. So there might be allele A3, there might be allele A4 also. Okay? So, but otherwise, typically in majority of the genetics, when people discuss, they talk in terms of in as two alleles are present for one gene, but there are possibilities of more number of alleles. So the dominant one is given the dominant trait or phenotype. The dominant phenotype is given that the first alphabet is taken as the capital R, and the small r is the recessive trait of that one. Here, why is it taken? The dominant feature happens to be round seeds. Okay, so the first alphabet is taken as the here, like this. And the small letter of R is given as the recessive. That means it is wrinkled. Small r would mean wrinkled seeds. Okay, you can see up here, that is how a round seed would look like, whereas this one, the wrinkled one, would look like this. So sorry, when you say capital R, that is homozygous, right? Both are same copies. R, small r, homozygous for wrinkled. And capital R and small r, it is heterozygous. Phenotype would be round, because that is the dominant character. Okay? Yes. Let's see what is happening here. I think this is simple, what you have already learned. Here you have taken a homozygous round and homozygous wrinkled. And then we can choose a cross has been made. I mean, how did Mendel perform that? He would have taken, say, anthers from one plant, I mean pollen from one plant, remove the pollen of other plant and put it on, make a cross fertilization. This is a flower, assume this is a flower. He would have cut off all the anthers so that there is no pollen grain. So you have the pistil and other things, ovules would have been here. He would have taken the pollen from here and then he would deposit on them. And that means you can understand how much work he has to do for doing these kinds of careful experiments for a large number of plants. There will be insects, there will be pollinators. I mean, those are the bees and other things. They are all pollinators. He has to take control of all those things as well. That's actually a difficult job. So anyway, so during gametogenesis, it will produce two types of gametes, R. If something has RR, and this is the cell, after meiosis, it will produce 4. One cell will produce R. All of them are same type, so here it is just shown as 1R. The same thing would have happened with the wrinkled plant as well. It is giving one type of gamete, and then hybridization, you will generate heterozygous plant. And heterozygous plant will have round, because round is the dominant phenotype. And then we have, this is called as the F1 generation or first filial generation. The previous one is called the parental generation, P generation. And if you had RR, what will it give rise to? A cell is RR. During upon G2 phase, I mean S phase and then G2 phase, it will become RR, RR, right? And then upon meiosis, it will produce R gametes with the ratio something like this. Capital R, and small r. So um, if there is fertilization that is happening, or you call it as inbreeding, or I mean you're mating, in this case you're mating um, these self-mating, self-pollination, okay, RR crossed with another RR. So you have two types here, RR, capital R, and then small r, small r. So we can just put as here capital R and small r, and here capital R and small r. And then you can, you know this is called the Punnett square, where you can arrange them R and R, small r and capital R, and capital R and small r, small r and small r. Always the dominant trait or dominant allele is put in the front, and then the recessive one. That is under convention. What you will find here is that you will have, there is a, what is the phenotypic ratio? What is phenotypic ratio? In this case, how many of them are round? It is, say, 1, 2, 3. 3 are round and 1 is wrinkled. What is the genotypic ratio? Sorry, genotypic ratio would be 1, that will be RR, is to 2 capital R small r is to 1 small r small r. So phenotypic ratio and genotypic ratio. This is what Mendel found. And every time he repeats this experiment, he will always come up with these kinds of ratios. And that is possible only if the segregation is independent. That is where the law of independent segregation comes. When you say independent and segregation, you have two alleles, R and R, and these are going into different gametes, and they would, upon fertilization, form a different type of genome. One second. Sorry. So these two alleles are segregating into the daughter cells independent of each other. If suppose there is a gamete, or if all the time these two RRs, two alleles of the same of a gene, are going together into one daughter cell, okay, then they are going together, they are not independently segregating, right? But what Mendel has, based on his ratios and genotypes and phenotypes, he has noticed that the two alleles of a gene are segregating independent of each other. They are not completely or always stuck together. That is the point of what is meant by independent segregation. Okay? And we have to remember these ratios. There is another feature that we have spoken about dominance, we have spoken about dominant and recessive. What this is codominance. Okay, one second. Okay, so in this case there is a red flower which is capital A. It is based on the pigment name. I think it is anthocyanin or so. And the other one is a white flower, which is small a. They would produce the homozygous for capital A would produce capital A gametes, and heterozygous one will produce small a gametes. Upon fertilization, they all produce pink flowers. Either if we spoke of only either red or red as dominant, we would expect here the genotype is capital A, small a. So we would expect that either the flowers are all red, then we would say red is dominant, or if all the flowers were white, we would have said white is the dominant. But that's not what is happening. In this case, what is happening is they are between these two, red and white, where red is also there and white is also there, and that gives rise to pink flowers. So both, this is a trait, red is a trait or phenotype, and white is a phenotype, and pink is a different phenotype, which is a blend of these two phenotypes, a and red and white. So that's where we call these two phenotypes as codominant. Okay, so that is one thing we have to remember, but otherwise the rest of the things will be same. Anytime you have a heterozygosity, you would find it pink. It's easier to determine the heterozygosity in terms of with regard to flower. It's easy because capital A has red color, small a has white color, and capital A, small a, the heterozygous, has pink color. We don't have to do much about it. Everything is evident. Compare it with the previous one, where we cannot determine what is the genotype of these, right? All these three, 1, 2, and 3, they all look round, but their genotypes are different. Compare it with this one. The homozygous capital A, homozygous small a are different colors. Heterozygous will be pink in color. Just by the phenotype, you can determine its genotype. Okay, this I think we have already, you read it, but I'll just finish it today and then we will see, we'll do problems on that in the next class. So dihybrid cross. I'm looking at, where is the other one? Did I miss? Okay, I'll continue in the next class, but here we'll say dihybrid cross. Di is for two hybrids we are making. That is about, you can think of hybrids of traits. If you're considering two traits in the same time, then you're working with dihybrid. So parental generation, F1 generation, and F2 generation. So here there are two traits. One of it is about whether the seed is round or not, and the other one is about whether the seed is yellow or not. Round is dominant, yellow is dominant. So the recessive one will be small r, that is green, wrinkled, and small y, that is green color of the seed. So when they produce gametes, they will produce ry, because they are homozygous. The gametes will be ry, or capital R, capital Y, and the wrinkled one, the homozygous, one second. The homozygous recessive one will make gametes of small r, small y, like this. And upon fusion, they would form capital R, capital Y, small y. And then we have 4 different types of gametes. The Punnett square will become complicated. And this is what forms. You'll have so many capital R, small capital Y, and the types of gametes will differ. I will only describe that and then we will continue in the next class. So I want to write the types of gametes this will form, right? The genotype of it is capital R, capital Y, small y. How many types of gametes can it form? It will be, in every, there is one principle is you have to try all the combinations. Every cell should get one allele for seed shape and one allele for seed color. So the combinations then will be capital R, capital Y, and it will be capital R, small y, right? This is from here to here, and these two will give like this. This one and this one will give rise to like this. And the second will be, the other one will be like this, small r, capital Y, and small r and small y. So their genotypes, if written, it will be like this, capital Y, small r, and small y. That is 4 different types of gametes can be formed. So when selfing or self-pollination occurs, if you are breeding F1 generations within itself, then you will have to make these. This is one set and this is one set, and now you're trying to see how many different diverse seed types might come up, and you will have a ratio on each of those. If you count the phenotypic ratio, so just the green, yellow is the dominant one. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. That will be, yellow is 12. And in that, you can again separate them, right? How many of them yellow are round? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 of them are round, and 3 of them are wrinkled. How many of them are green? 1, 2, 3, 4. 4 of them are green in color, out of which 3 are 3 are round and 1 is wrinkled. So what is the phenotypic ratio then? It will be 9 is to 3 is to 3 is to 1. What are those 9? 9 are yellow, round. And this 3 refers to yellow, wrinkled. And the next 3 is green, round. And there is one that is green, wrinkled. Okay, that is the phenotypic ratio of in a dihybrid cross like this. We'll discuss the next things in the next class. We will discuss about little bit basics about mapping maybe, and we will talk about, we'll continue the genotypic ratio, and we will do problems mostly. And I encourage you to watch the video some come, so that we can spend more time doing some problems. Okay, take care, and we'll see you. Any questions?